This project is a collaboration between myself and Matthew Harris. And I just like to acknowledge up front the people in our groups who've actually contributed the data so that I don't run out of time and forget at the end. And so this is a former postdoc in my lab, Michelle, and my current technician, Alexandra, who've been generating and doing a lot of the data processing in my group. And these are people in the Harris lab. I know Katrine is a postdoc and Brent is a student. I actually don't know Mira, so a technician, okay. And I'm going to be doing most of the talking, but Matthew is right over there. So any detailed questions about his data or their experiments, I'll direct you over to him. I would say that one of the primary goals in the spirit of contributing to the community, one of the primary goals of our project is to generate a user-friendly online database that gives you detailed anatomical information about the formation and development of the zebrafish skull. And ultimately, we would like this to be accessible to not just zebrafish researchers working outside of FaceBase, but also the broader community, so people working in other model organisms, especially mouse, since that's primarily the organism that's being used to generate genetic models of human diseases, and ultimately also researchers working with human patients who want to, for example, compare phenotypic features of a human disease to an equivalent in zebrafish or in mouse. And we have two primary methods of generating this data that I think are very nicely complementary. And so in our lab, we primarily are doing imaging of transgenic zebrafish expressing markers of different cell populations. And in the Harris lab, they're using high-resolution micro-computed tomography or micro-CT. And so first I'm going to go through some of the examples of our data and talk about the strengths and limitations of the two approaches. And then later, really I'd like to focus on what we're trying to do to better integrate our two data sets and also really make them very useful to the broader community. And so I hope we can get a discussion going about the best ways to do that as we go through this. So initially, we have been doing all of our imaging based on these two transgenic lines. They are particularly useful. So the collagen line marks is a general marker for cartilage. The SP7 M-cherry line is a general marker for osteoblasts. And they're particularly useful for this type of imaging because they're very bright, they have almost no background, and they maintain a high level of expression into adult tissues. So as long as we can get, our confocal to penetrate into the tissue, we can get nice images. And we do all of our imaging on a what Leica calls a macro confocal microscope. So it's basically the laser and detection system of a standard confocal, but using the optics of a high-end zoom macroscope. And this gives us a tremendous working distance, allows us to handle extremely large samples, gives us good penetration into the tissue, and gives us the flexibility of going all the way from extremely small to extremely large samples. And because we're doing confocal imaging, of course, we're generating stacks of data. And so we can zoom through the Z stacks to look at different levels of expression. And we can also do 3D reconstructions and generate models that we can rotate and look at from different angles. Of course, the limitation of one limitation of confocal imaging is that you never get quite as good a resolution in the Z-axis as you do in the X and the Y. And, this is really a practical limit of how long you can keep samples on the microscope and continue to scan, and, how large of a data set are you going to generate, and how long can you keep your fish alive. So we're doing, I didn't emphasize that at the beginning, but we're doing our imaging primarily on live samples. And so although, we're somewhat limited by resolution, we have a tremendous advantage of being able to do dynamic imaging on individual fish. And so this next data set illustrates one of those sets of data. So these are just the, projections of the Z stacks from a single fish going through this period where most of the skull bones are forming. And I want to emphasize that this is, I sort of feel like I'm underplaying the data by just putting it all on one slide. This actually took a tremendous amount of effort on Michelle's part to figure out how to get an individual fish to survive and tolerate being re-anesthetized every couple days through this entire time period. But she spent a lot of time working this out, and once we sort of got the method going, we've been able to generate a very large number of data sets, mostly of wild-type fish, but we've started doing some mutants. I'll show you a little bit of that in a minute. And again, mostly with this particular double transgenic combination, but we would like to start expanding that to look at some of our existing transgenic lines that highlight other cell populations or the expression pattern of other genes. And so I think that in future years of the grant, that's a really good opportunity for expanding this data, is to think about intelligently about additional transgenic lines that we want to generate, and how we can effectively compare the data among the different lines once we sort of get it out there and onto the website. And we've also been able to start, because we've done a fairly large number now of, especially of wild-type fish, and we're starting to accumulate more data on the mutants, we can try to get quantitative and measure things like the growth of specific bones. So this particular graph shows the growth in area of the frontal bone during this time period, and it just illustrates, some things that weren't obvious to us when we just first looked at the data. And in particular, we find it interesting that there's sort of a fairly long lag time of slow growth here between initiation of the frontal bone and when you see this sort of rapid takeoff and acceleration in the growth. And so, I don't know if we would have seen this that easily if we hadn't kind of analyzed the data in this way. And now we can think about what's happening biologically at that transition, and, how the behavior of the cells is changing to go from this very, slow growth period to the really rapid planar growth. So in the Harris lab, they're using a high-resolution micro-CT to do imaging on whole mount specimens. And this is just a poor little fish stuck in the tube here, being rotated around. But they can generate really beautiful high-resolution images of these fixed samples. And unlike confocal imaging, the micro-CT is equivalent resolution in all axes. And it lends itself very well to, okay, my movie is not playing. Oh, no, I'm sorry, Matt. So the other movies were playing. I don't know why this one's. Anyway, you could see, if you could see this movie, it's a 3D model of this data that, you can rotate and look at from different angles, but also do in silico sectioning to look at different planes of the data in the samples. And, so this is the kind of data that will be, or may already be, I'm not quite sure, available on the website. And so I think even just the raw data can be extremely useful for the aficionados in the field who are, really into skull development in zebrafish. But as I said, eventually we want to make it more useful to the broader community and people who don't necessarily have our level of detailed interest and knowledge. Now, one of the primary limitations of the micro-CT imaging, of course, is that it's based on mineralization in the bone. And so there's definitely a lower limit at which the calcification that's naturally occurring in the bone just simply doesn't allow visualization. And so you can see that obviously in this image of a young juvenile fish. While we know at this stage by both looking at the transgenic lines and looking at, with a more sensitive technique like alizarin red staining, that there's a fair amount of bone already present, but you simply can't visualize it with the micro-CT. So in Matt's lab, they've been working on enhancement methods to allow efficient or effective imaging of younger samples. And so they're using a whole mount staining method based on silver nitrate. So this is the primary compound in von Casa histological staining. And it in whole mount is as sensitive as alizarin red at showing bone in small specimens. And because it's electron dense, it enhances also the micro-CT. And you can see here the incredible difference it makes. So this is the same individual specimen before and after the silver nitrate staining. So we're really getting a tremendous enhancement of sensitivity using this method. And so between us pushing the upper limit of the confocal imaging and the Harris lab pushing the lower limit of the micro-CT imaging, we're managing now to meet in the middle with some of our specimens. Oh, sorry, and I just wanted to mention that although I would say the silver nitrate is completely necessary at the earliest stages, but even at slightly older stages where it's feasible to do the micro-CT, it definitely enhances the imaging, and you can see that in this developmental series without and with the silver nitrate. So as I said, now we can start to meet in the middle, and so several times now we have taken one of our fish through the imaging protocol and then at the end fixed the specimen and sent it over to their lab, and they've done the micro-CT. And so this is one example of a wild-type fish, and I think this was at about 30 days post-fertilization at the end of our imaging. And so then we passed it on to their group. They did the silver nitrate enhancement and got a really nice micro-CT image of exactly the same fish. And so now that we can generate this data on the same specimens, we can start to think about issues of overlapping that data in a practical way and making it possible for people to look at both things in the same samples. So as I said, we've done most of our imaging on wild-type fish. We've started to do some mutants. This is one example that we started to work on early on because this is the Zebrafish Chihuahua mutation. It's a mutation in one of the type 1 collagen genes that's a really good model for human osteogenesis imperfecta. And so you can see here in the adult fish that there are obviously a lot of problems, not just with the skull, but with the skeleton in general. But the larvae look normal. And so this is kind of a big black hole for us of when you first start to see the problems cropping up in skull development. And so we've been trying to fill in that hole by doing some more imaging. And this just shows a small sample of some of the data that Michelle generated. And one thing that's come out of this is that although we've standardized the imaging, not just, so these are, I'm listing the days post-fertilization, but actually when we upload the data to the website or to the hub, we're standardizing everything by standard length. And so even at equivalent standard lengths, the growth of the cranial vault is lagging behind the growth of the wild type skull. And so this is even though the rest of the skeleton is basically the same at this stage. This is raises some interesting questions about the biology and why you see this lag. But it also is allowing us to kind of generate a paradigm for how to compare other mutant phenotypes as well, as we either our groups or other groups start to generate these mutants, and we can apply some of the same imaging techniques and try to be really quantitative about comparing phenotypes. I'm going to skip over that slide. And then this is just, again, at the end of this experiment, we fixed the fish and sent it over to the Harris group, and they did their magic and generated the micro CT image of the same fish. Okay, so another enhancement of our data that we've been doing a lot of talking and thinking about is how to make it easier to visualize the complexity of the zebrafish skull. So there are a far greater number of individual bones in the zebrafish skull than in either human or mouse. And it's, obviously a very complex structure and maybe not so accessible to, kind of think about and visualize. So we want to make that easier. And one good way to do that is to segment the data. So in other words, to computationally separate individual bones and other elements. And then once you've done that, you can do a lot of things with that information. And so Matthew's been working on this with his micro CT data. And just to show a couple examples, you could say color code the individual elements according to their embryologic origin, either neural crest or mesoderm, or you can separate them out by regional sort of anatomical regions in the skull. And you could make every single one of those 100+ bones a different color and, do a lot of in silico manipulations of this. So we're also starting to work on how to do this with our confocal data, which has slightly different challenges because the nature of the data is a little different. But using this is one example of using the segmentation editor in ImageJ, being able to isolate a single cartilage element from the data in the skull at this stage. And this is the epiphyseal bar cartilage. And so you can generate this three-dimensional model and similarly, color code all the different regions and put them all together into a single overall model. So, we would like to be able to accomplish what, Yang and his group have been able to do with the mouse data. And so similarly to building a mouse, we want to try to build a fish. And that, I think this would be a really user-friendly, very accessible way for the broader community to look at the data that we're generating. So a few additional issues that we've been thinking about, and I would welcome, questions or input from other people. One thing I've thought a lot about is registration between different data sets. So, for example, we've generated expression data, and then, so two examples. One, so we've generated expression data on one individual fish across time, and is there a way that we can register those individual data sets and sort of generate an overall movie of some kind that takes you through the stages of development in one sample? So that's one issue. Another issue is taking two different types of data, so taking the confocal imaging data and the micro CT, and then being able to really line those up and make sort of one data set that contains both sets of information. Ultimately, I didn't really point this out, but so far we've been doing all of our confocal imaging basically from the top down, and we're somewhat limited by the tissue depth and how far the light can penetrate. And so I'm trying to think of ways that we can do sort of the equivalent of the rotating micro CT. So we could, turn the samples over, acquire data from the other side, and then be able to register those two data sets to create one overall picture of the entire head in 3 dimensions. So these are some of the things that we're thinking about in the short term. And I think at that point I will stop and if there's a little bit of time, take questions or comments. Yeah.
So I was wondering how much variation that you're noticing between individual fish, since you're looking at days post-fertilization versus hours, because you would imagine in hours you might see more than days.
Right. So that's a great question, and that's why, although I put days up here because I'm being a little bit lazy, it's easier for us to just say that. But in reality, for all of those fish, we're measuring the standard length. And so you see, if you used days as your measure, you would see a lot of variability. When you use standard length, you see much less variability. There's still some. And I apologize, I should have put that data in for for example, the measurements of the frontal bones. And actually, the Harris lab has done, I would say, a better job than we have of generating larger sort of quantitative data sets and measuring different aspects of the skull development. But when you standardize by standard length, you actually don't see that much variability. I would say in our hands, at least with the mutants that we've looked at so far, we see more variability in the mutants than in the wild type. And it actually kind of also raises another interesting question. So with the Chihuahua mutant, the overall fish is shorter. And so you don't see that so much at the beginning when they're younger, but as they get older, that difference gets greater between the wild type and the mutant. So then is standard length really a good way to normalize that data? And it probably isn't, but we haven't come up with a better way yet.
Yeah, I mean, I would say for the wild type it probably is, but maybe the mutants, I don't know.
Yeah.
I don't know what the best way is. Anybody else?
So I have actually done quite a bit of image registration and actually have done things right on younger fish where you image from the top and the bottom. And you can register them pretty well.
Okay, great. I was hoping I wouldn't have to reinvent the wheel.
No.
I figured somebody knew how to do it. I just hadn't figured it out yet.
It's been a while, but I think I can probably figure it out again. And also, I was going to say is that I think that mapping your gene expression onto the micro CT is going to be really useful, and because you have isotropic resolution with the micro CT. But I mean, I would actually probably think about taking it from 4 or 5 different sides, or 4 sides.
Well, we were actually we've been starting to try to do that. Yes. So and I think our problem, my technician was a little ambitious, and she started with fish that are too big. So I said, let's start with the low-hanging fruit first.
And you may, if you can, see if you can find somebody who's got access to a light sheet. There are light sheets that'll do about the same, that scale.
Yeah. No, I know. That would have been, I wish I knew somebody. Actually, I don't know, Matt, is there somebody in the Boston area who has one? Okay. Yeah, so that's a possibility also. Although not for the live imaging. Oh, yeah, okay. Yeah, I guess you're right. Maybe at least for the younger stages, it'll work for live imaging. Yeah.
Okay, great. Well, let's give Shannon another hand, and Young is going to be up.